Why did the chicken cross the road? Evolution, how did this behavior evolve, why its adaptive offspring? Molecular genetics, actual genes control the evolution and how it happened. Behavior genetics, individual genes that produce the variation. Ethology, studying behavior in nature, fixed action pattern, stimulus. Neuroscience. Brain. You get an input coming in of some stimulus, that makes the chicken cross the road. What was going on in the chicken's brain a few milliseconds before to cross the road? Why specific cells in the brain are doing this? And how are they doing it? Overview of the brain and the nervous system. How different parts of the brain are specialized for different behaviors. The actual cells in the brain, how they communicate. The chief function of the body is to carry the brain around. Thomas Edison. But it can be very brain-centric. How different parts of the brain control different behaviors without considering bigger evolutionary aspects. Brain and nervous system. Nervous system, 2 classes. 1 central nervous system. 2 peripheral nervous system. 1. Brain and spinal cord. Brainstem sits at the bottom of the brain, at the top of the spinal cord. It's relaying information to and from the spinal cord, to and from the brain. Your brain doing the processing, and the spinal cord sending the signals and sensory information. It regulates what goes through. Cerebellum, sitting at the very back of the brain, very wrinkled, different cells packed into it. It controls your motor movement. Helps to learn something new. It helps correct the mistakes. Cortex, 4 different lobes. 1 frontal lobe, at the front of the brain. Plans your actions. Controls your movement. Different parts of the frontal lobe, that control movement, control different parts of your body, it's organized according to function. 2 parietal lobe. It's right next to the frontal lobe, which senses the sensory touch information. Different parts of the parietal lobe are specialized for different parts of the body. Also, the size of the cortex that receive that information are also different sizes. 3 temporal lobe, below the parietal lobe, receives the auditory information. Deep in it, the parts of the brain that help memory formation. 4 occipital lobe, back of the brain. Visual information. Limbic system. Structures underneath the cortex, sort of above the brainstem. Emotions. Learning. Memory. Divided in hippocampus and amygdala. Hippocampus, seahorse. Memory. Forms new memories. They found this part of the brain by accident. Patient He had terrible seizures, very bad epilepsy, incontrollable. They figured out where the seizures started, and surgically removed that part, which included the hippocampus. They took out the hippocampus on both sides of the brain. So, he didn't have seizures anymore. But he couldn't form any new memories. And if you ask him for memories from his childhood, he could still remember those. Amygdala, sitting up in the front of the hippocampus. Fear and anxiety. When you're smelling the sweat from a scares person, the amygdala lights up. Hypothalamus and pituitary gland. Hormone central. How hormones are released to the rest of the body. Control behaviors. Hypothalamus sits in the center of the brain at the very bottom, and the pituitary is underneath it. It controls the 4 Fs. Fight. Flight. Feeding behavior. Reproductive behavior. Spinal cord. Parts that send out information, other parts that receive information. So, you've got motor nerves and sensory nerves in different parts of the spinal cord. Peripheral nervous system. Has the motor nerves and sensory nerves that are outside the spinal cord. Like touch information or heat. Also, a whole part of the peripheral nervous system happens automatically. Like heartbeat, digestion, breathing. What's actually inside a brain? Cells, what they're doing, how they make you behave. Santiago Ramon y Cayal. 1890s. The general theory about the way the brain worked was that it wasn't individual cells performing their functions. They thought it was an interconnected web. They only knew that the brain was where behavior started and where it was controlled. So, he found a technique invented by Golgi. He took small slices of the brain and turned 1% of the cells in that slice black. Showing that individual cells in the brain are doing the work. So, 90% of the cells in your brain and spinal cord are not actually neurons. Called glia, it means glue. Though it was just glue sticking the neurons together. But that's not the case. Astrocytes are star-shaped cells. They supply nutrients to the neurons and regulate how they fire. Oligodendrocytes or Schwann cells. They wrap around the wired of neurons. And make their firing go faster. Microglia. The brain's immune system. They send little processes into parts of the brain to see if the brain is getting infected by viruses, bacteria, dead cells. All these 90% are doing this, so your neurons work. Neurons. Computational units of the brain. You've got about 100 billion neurons, and each has about 10,000 connections to other neurons. Connections are called synapses. Then you got a quadrillion synapse. Compare that with the stars we can see in our galaxy, it's only about 300 billion. How does it actually work? Neurons have dendrites, receive the information on a neuron, from the cell before it, and pass it on down into the cell. Soma, the cell body of the neuron. Nucleus, has all the DNA in it. Now, the cell has to decide whether to pass that information on to the next cell or not. And that happens in the axon hillock. Axon, the wire sending the information onto the next cell. Terminal, where the cell will send on information onto the next neuron. How it sends that signal? You have a cell, that's either on or off. It keeps off until it's ready to send a signal. Neuron keeping a resting potential. How does a neuron do that? The way neuron sends information is through movement of chemicals called ions, which are charge atoms. They have a positive or a negative charge. So, the neuron has to be quiet. Does it have positive ions outside of the neurons? Because the flow of the ions is going to communicate the signal. It keeps these ions out of the cell by using pumps. So, it has pumps that will pump out positive charges from the inside of the cell to the outside of the cell. That means that the net charge on the inside of the cell is negative. This is how the neuron stays quiet. How does it make it fire? You have another neuron, sending a chemical signal. That chemical signal is going to get received at the dendrites. When that chemical called a neurotransmitter, hits a receptor out on the dendrite, that receptor is going to open up a channel, and that channel is going to let some positive ions pass, is receiving a signal. So, you get a change in charge. The neuron is now getting more positive on the inside, before it was polarized a big negative charge on the inside, and now it's getting depolarized. Then, the neuron has to decide if to fire or not. But from only one signal sent, you might not get a lot. Now, imagine you're getting a lot of frequent chemical messages, getting lots of positive change into the cell. Or getting several different messages at the same time. Then, you end up with enough positive charge inside the neuron, at the axon hillock, to make the neuron decide to fire. So, what makes it decide? There are more channels in the axon hillock, that can open up to let more positive ions in. And they decide whether enough positive charge. Once that happens, it's going to feed forward. You're going to get more positive ion going in plus more channels opening equals neuron sending a signal. It keeps having positive ions go in, all the way to the axon, all the way to the terminal. Then when you get positive ions into the terminal, it's going to send a chemical message onto the next cell. And start the whole process again. The most important thing is that the decision made in the axon hillock is all or nothing, there's no turning back. Called action potential. So how does it stop? You have other positive ions on the inside, right after it opens. You can get the cell opening other channels, to let the positive ions, a different kind of them, flow out. Then you get restoration of the balance and charge. Also, you have the pumps, that continue to send the positive ions out of the cell. In short, different parts of the brain do different things. Like in the spinal cord, different parts function for different behaviors. You've got different cells, with different function in the brain. Neurons. Glia. Different type of neurons. Neurons are individual cells making decisions, they're the functional unit of the nervous system. And when they send that signal, it's an all or nothing process, action potential. 1800s. Neurons were thought to be connected by cytoplasm. And it was until the advance of microscopy and staining. In 1891, H.W. Gottfried. Neuron doctrine. That this network was made up of individual cells. So, if you have separate neurons, if one is to communicate to another, it cannot happen by electrical means only. It must happen by chemical means. So, before the events at the synapse can take place, we need to go back to the action potential, which starts at the axon hillock, where to fire or not the action potential, once it's fired will go to the axon terminal. Now, the axon terminal is a structure that stores a lot of neurotransmitters, they're packaged in membranous spherical structures called vesicles. So, when the axon potential reaches the axon terminal, it will trigger an influx of positive charge, which will release neurotransmitter from these vesicles. Terminology. Signaling neuron equals presynaptic neuron. Neuron that receives the signal equals postsynaptic neuron. Junction at which these two neurons connect and communicate equals synapse. So, once this neuron is bound to the receptor, it can trigger two temporal effects. One immediate effect, the opening of a channel, which allow ions to jump in, could be either positive or negative charge. If they're positive, it will persuade an action potential to happen if there are enough charges. If they're negative, it will dissuade. 2. When a genomic effect is induced. A neurotransmitter might be released and might bind to a receptor, which might influence the activation of a transcription factor, which might induce the production of more receptor channels, that might find the way onto the dendrites. So, if you produce more channels or receptors, you make the synapse more responsive to the same amount of neurotransmitter, strengthening the synapse. So, a single neuron can respond to different types of neurotransmitters. Could be inhibitory or excitatory. It's also possible for a single neurotransmitter to have an effect on multiple neuron types located in different areas of the brain with different functions. The brain having different functions in different areas. Because different networks of neurons are responsible for different functions. They have a physical separation between different networks. So, it's possible to use a single type of neurotransmitter because of this physical separation. Compartmentalization in the brain. Because of this redundant use of neurotransmitter, you only need a few hundred. What evidence you need to provide to prove that a molecule is a neurotransmitter? Where are neurotransmitters located? They're located in the axon terminal. What triggers the action of a neurotransmitter? You need to prove that the release is following an action potential. What's the effect of a neurotransmitter? You need to prove that after a neurotransmitter is released and it binds to the receptor, it induces an influx of charge in the postsynaptic dendrite. Some neurotransmitters. Dopamine. Reward system. Pleasure. Also, remembering the previous. One type of neurotransmitter in an organism may have different functions. So, all of these has a lot of functions. Epinephrine, adrenaline. Fight or flight response. Norepinephrine. Is pretty much interchangeable with epinephrine. They share similar a structure. So, you can change epinephrine to norepinephrine and vice versa. Serotonin. Sleep. Appetite. Mood. Acetylcholine. GABA and glutamate. Most common neurotransmitter in the brain. GABA, it's the most common inhibitory neurotransmitter. Glutamate, it's the most common excitatory neurotransmitter. Neuromuscular junction. You can't behave unless you're able to move. Movement lies in the contraction of muscles. So, a synapse will occur on the nerve that signals to the muscle, which will release the neurotransmitter, acetylcholine. It will bind to the receptors in the muscle, which will trigger a contraction. Neuromuscular junction only uses one type. Neuropharmacology. External manipulation of synaptic events. Maybe for research purposes. Or to correct disease states. To increase or decrease the strength of communication across a synapse. Like the hallucinogenic. Such as mescaline, LSD, psilocybin. These interact with serotonin receptor, because they have very similar structure to serotonin. How to strengthen the synaptic response. Increase the release of neurotransmitter from the presynaptic neuron. Like increase the synthesis of neurotransmitter. Force the released neurotransmitter to linger in the synapse. You can block reuptake or degradation. So, when a neurotransmitter has done its job, has bound to the receptor, is used up. You can't leave it in the synapse, otherwise will keep signaling to the receptor. So, you can use two ways. One, reuptake, reinjection of a used up neurotransmitter back into the presynaptic neuron. Like, a protein pumps the neurotransmitter into the presynapse. Or a protein to repackage the neurotransmitters in the vesicles. 2. Degradation, degrade used up neurotransmitters, which can be detected in your cerebral spinal fluid, or blood, or urine. Useful to detect levels of neurotransmitter to diagnose diseases. Neuropathology. Something wrong with the brain. Like Parkinson. They have an insufficient or decreased level of dopamine in the area of the brain that controls motor movement. So, if you want to increase dopamine levels in the brain, you might alleviate the symptoms of Parkinson's. But you will increase them in all the brain. So, you might change levels in the mesolimbic pathway from normal to too high, as a result you'll induce symptoms of schizophrenia. So, the brain isn't really compartmentalized. Because if you move something from one network it can change another one. Which result in deleterious side effects in other areas. In short, the process of action potential moving along the axon, and how it influences the influx of positive ions at the axon terminal, which will release neurotransmitter. Neurotransmitters after they're used up can be degraded or recycled, reuptake, and that degraded products can be detected in blood, urine, and cerebral spinal fluid. Compartmentalization. Pharmacological manipulations, you have to be careful, especially when something is wrong in only one area of the brain.